Hi guys! So today we're going to be talking about chapters 3.1 and 3.2 of topic 3, which is genetics in bio. So first, we're going to talk about a few keywords that you're going to need before going on your test, which are going to be karyotype. We're going to see that later. Diploid and haploid cells. So a diploid cell has two N chromosomes and a haploid cell has N chromosomes. So N is just the number of chromosomes. So let's say a haploid cell has 13N. N is 13. A diploid cell will have 26 chromosomes. So we're going to see these in our meiosis topic, which is 3.3, which is not in this video. I can make one though. A gene is a segment of DNA that codes for the synthesis of a specific polypeptide. An allele is a different version of a gene. So let's say a gene is going to be the coding of a polypeptide, which is the tall gene. An allele is going to be a different version of this gene. So it's not going to be a tall, it's going to be a short. So that's kind of the relationship between gene and allele. It's just a different version of a gene. That is a definition of allele. Gene locus is you can see from here, it's the location of the gene on the chromosome. So let's say you have a chromosome here. You have a gene here. This will be the gene locus. It'll be at the top of the gene. Okay. Centromere is we should keep that chromosome. Centromere. So the centromere is the location in the middle of both. Let's say this is one chromosome. And this is the copy of that chromosome. These are, okay. The location where they're both connected in the middle is called the centromere. Okay. A homologous chromosome, we're going to see this soon. So I'm not going to go into that right now, but I hope that clears it up. Okay. First, let's start talking about DNA. Well, DNA is the genetic material that is in our bodies. So what really is it? It's this string-like material made out of nucleic acids and other stuff like that. And it's wrapped around these proteins, which are called histones. Histone proteins. This is essential for you to know because it kind of allows us to contrast the difference between a prokaryote and a eukaryote, because prokaryotes don't have these histone proteins. Only eukaryotes do. So that's just important to know. Okay, so now we're going to be going into a little bit about homologous chromosomes. Homologous chromosomes. So what are homologous chromosomes? There are two chromosomes that have the centromere in the same spot first. Centromere positioning. Which is this. This is our centromere positioning. They have that the same. Another characteristic about homologous chromosomes is that they have the same banding pattern. Banding pattern is basically the genes that are located in the same places. So let's say this gene and this gene have different alleles. They're located in the same place. This gene and this gene are coding for both coding for, let's say, I'm trying to think of something that's not polygenic. Okay, anyways, let's say one of them is for eye color. So these both code for eye color. That's not how it works, but that's how it codes for things in the same spot. Third, they are the same length, which I think is very obvious by this drawing. Same length here, same length here, these two, and these two. These three you have to check them off before you determine that they are homologous chromosomes. Same centromere positioning between these two? Yup. Okay. Same banding pattern? Okay. Yup. Same length? Yep, okay, they must be homologous chromosomes. These are also important for meiosis. Okay, next, moving on to karyograms. So karyograms are basically the image of all the chromosomes in our body. It's the image of our karyotype. So the karyotype is just the it's the chromosomes in our body. It's like the genome but for chromosomes. So it's called a karyotype. It's each person has an individual karyotype. But when you take a picture of this karyotype and under a microscope and organize the chromosomes from decreasing length, it's considered a karyogram. So it's important to note that these chromosomes are organized in decreasing length. We can see number 1 is the largest and number 22 is the smallest. So these are for the autosomal ones. Autosomal means there are the regular body chromosomes. They're not our gametes, they're not anything like that. So those are the yellow ones. Yellow is autosomal, which are pairs 1 to 22. Okay, so these, as we said, are organized in decreasing length, and each of these are homologous chromosomes. So we take this pair, for example, these over here. They're homologous chromosomes. We can see they have the same orange spot in the middle. That must be the centromere. They have the same banding pattern. Yes, they do. And they are the same length, which all indicate that they are homologous chromosomes. Okay, great. Now, let's look at the last pair of chromosomes, which are these. The last pair, which is the 23rd pair of chromosomes, are called the sex chromosomes. These determine if you are a male or if you are a female. There are lots of chromosomal abnormalities that result in having an extra X or a Y with 2 X's. These are all chromosomal abnormalities which are happen at birth, during meiosis. These are all meiosis things, which should be in the meiosis video. Yeah, okay, so that's what you need to know about those chromosomes. And yeah, now let's take a look at this karyotype. This is a special karyotype. Take a minute and try to notice something special about this karyotype. I'll let you guys discover that. Remember, we have to look at the number of chromosomes. So from a karyotype, we can identify the number of chromosomes. We can identify the structure of chromosomes. And yeah, these are the 2 main things we can notice. Okay, so what we can notice about this karyogram is that there are 3 chromosomes in the number 21. Right? Look, all of these have 2. 1, 2, 1, 2, 1, 2. This one has 3. So this is actually a chromosomal abnormality called trisomy 21, which can also be denoted as Down syndrome. This is a occurrence that happens in meiosis, of course, and this person has an extra chromosome, which is kind of crazy, which results in Down syndrome. Not Down syndrome, it's Down syndrome. Okay, so see how we can tell from a karyotype? We can tell the number of chromosomes. If it's abnormal, then we can tell that this child is going to have Down syndrome. That's why karyotypes are very important when before having a child. Okay, so how do we get these cells for the karyotypes? Well, while we take this karyotype, we try to take the karyotype of the baby using a karyogram while the baby is still in the womb. So how do we do this? The first method to do this is amniocentesis. So amniocentesis is when there's a, say this is a pregnant woman, and this is her, that's a horrible photo. This is a pregnant woman, and her hands are on the wrong spot. So they take a needle and they put it through her, and they take some amniotic fluid, which is the fluid that's found in the womb, and they take it out and then they test it and then they make a karyogram. That is amniocentesis. The nuchal translucency scan is when they do some kind of an ultrasound scan. So it's more like a non-invasive kind of thing where they see here, they see different locations. Let's say this is the baby. And they see the amount of amniotic fluid in one of the parts of the they find the nuchal fluid. And if it's a large amount, they correlate it with Down syndrome, and if it's a small amount, they correlate it with a healthy child. Last of all, we have the chorion villus sampling. So in a womb, in the stomach of a pregnant woman, there is the placenta, which is what connects the uterus to the wall of the stomach, the placenta. And so what they do is they take a needle, I think, and they go into the placenta, the part of the placenta where the baby is linked to, and they take a part of the placenta out. This is the baby part of the placenta, which I believe is called the chorion, which is why it's called chorion villus sampling. And they test for it. And they try to see if they do the karyotype and they see if it has Down syndrome. Okay, well, that's basically it for 3.1 and 3.2. Let me know if you want another video. Okay, bye.